Okay, a little bit about my background. I don't like to talk too much about my background because it's a lot. My original background is in biology. I'm from Brazil originally. I've been in the US this time for 9 years. I did part of my PhD in Kansas and then my postdoc in New Jersey. So my PhD is in developmental biology and tissue and cellular biology, and my postdoc is in molecular biology, things that we can't see but we believe that they're there. So I worked in Germany also for WHO and the Rockefeller Foundation in a very interesting project involving molecular biology, and then I moved to New Jersey for my postdoc, went back to Brazil, taught at universities for medical students and all the medical area classes. And for personal and family reasons, we decided to move back to the US, and we've been here for 9 years. I've been with Asea for 3.5 years. It'll be 4 years in March. So I joined Asea right before COVID hit in March 2020. It was interesting because I came to work. I was hired first to work at the regulatory department. So I was working, and then suddenly we couldn't come to work anymore. And I'm like, am I gonna be fired? Because it's weird, right? You have to work, but not work. You work from home. This entire concept was kind of still different for me at that time. Nowadays it's very common, so yeah, so I'm with Asea for almost 4 years. It's been a pleasure working here. When I first joined Asea at the regulatory department, I was kind of skeptical about all the things that they claimed about the water and so on. And as I kept studying it and relating the water, Renew 28 and the redox molecules, and I saw the results, I saw the studies, and I myself then was, not myself, but I was invited to go to R&D and start doing the science behind all these products. I understood better the products, and now I'm like in love with all of them. I 100% believe what I'm going to talk about because I've seen it, I studied it, and we've seen results one after the other that prove scientifically, like things that we can see that is happening, and it's 100% backed by science. Everything that I'm going to talk to you today. So let's understand first what redox is. Redox is a word that was created because of these two other words. It's short for reduction and oxidation. And what does that mean? This is a nice, and I like this quote a lot, saying that life is nothing but electrons looking for a place to rest. Because redox, reduction and oxidation, is all about electrons and the movement of these electrons. So what is an electron? Electron is a very small particle that has a negative charge. So we remember, we hope you all remember that from high school, right? We had like positive charge, neutral charge, and negative charge. Electrons are the negative charge. They're very, very tiny molecules, and they can move almost at the speed of light. And redox is about this. It's about electron transfer, which is a process that basically involves the movement of electrons from one molecule to another. And when they move from one molecule to another, they're always looking for a place to rest. What does that mean? It means that they're looking for, because electrons, it's basic energy. So they're looking for a place with less energy so they can rest. They can be in peace. They don't have to keep moving. So that's what the redox reaction, that's when the redox reaction happens. They happen when these electrons are looking for a place with less energy, a place to rest. So if they are in a molecule with very high energy, it's like a ball, for example, in the top of a hill. What's gonna happen if we push this ball to the edge? It's gonna roll down, right? And why does it happen?
Finds place to rest.
Perfect, yeah. Because it's complicated to keep like being there, right? So they roll down to find a place to rest. And this is the same, and this is how the energy goes from, or the electrons go from high energy to low energy. This is just an image about oxidation and reduction. So the movement of electrons from a high energy place to a lower energy place. This place is obviously a molecule, an atom. Okay. Any questions?
I missed the second part, like this is an atom. Okay, so reduction is gaining the electron, oxidation is giving. Okay. Thank you.
Okay.
Hunter has a really nice way for you to remember that.
Yeah,
Called
he tells oil rig. Oxidation is losing, reduction is gaining electron.
Why does that make oil rig?
Oxidation is the O.
Oil rig.
I is
So oxidation
Is losing. Okay, I get it.
Reduction
Reduction
is
I
gaining.
love that. Thank you.
That is easy.
Yes.
Thanks, Jerry. Okay, so finally we come to our signaling molecules. How do we do that? How do we do these molecules? These molecules are based on redox reactions. So you've been to our plant, right? So you see all those big barrels, big things where we have the molecules, the water and everything. But how is this process? How the process happens? What is going on in there? So if you read the label, we'll see that in there, it's written there's water and salt, right? What has happened when we mix salt and water? What happens to the salt?
It dissolves.
Dissolves. Why?
lower energy state.
Yes, it's easier to dissolve than keep like as salt, right? So they dissolve. It's a reaction. So we get to this state where we have a saline. Okay, so this is where we stand before we apply electricity. So to produce signaling molecules, we get this mixture here and we apply energy. That's the pattern process that we have. And we'll push this resting molecules into a state that they will be, they will have to react because there is electricity, energy, pushing them to some reactions. And then it will be created the redoxing molecules. Okay? This is our process, and this is how we create the redoxing molecules into those barrels that you saw that there's all like the aluminum in the outside. Okay? Okay, so when we create these molecules, the signaling molecules, we understand and we know that these molecules are very, very tiny molecules. And this is part of the products that we know as Asea Redox Cell Signaling Supplement, the Renew 28, do we have it here?
Yes.
Yes, Renew 28, the Redox Clay Mask, and the Intensive Serum. So these 4 products at Asea, they all contain redox signaling molecules. And how do they work? These small molecules. So we postulated this this way that these molecules are probably working, and we've been working on research to prove that, and we've been seeing that this is mostly how it works in our body. So they are tiny molecules. Tiny molecules in our body, they do not need permission to enter our cells. They are just absorbed. So that's how the signaling molecules are first in contact with our body. They are absorbed. When a molecule is absorbed and goes into the cells, they will trigger something. They will trigger a pathway. They will play some role in the cells. In this case, our cells, and we'll see that we have studies showing that, these molecules will turn switches in the cell that will turn on and off genetic pathways. They will regulate some genes. This regulation will have a consequence. The consequence of this upregulation or this modulation of genes is the production of certain proteins, right? These proteins will be sent to outside of the cell and will act in other cells around it. So it will produce a cascade effect. So from one cell to the neighboring cell, this cell to the neighboring cell, and this is like a domino effect.
I have a question.
Yes.
What prevents it from like turning on a bad gene, like ERT?
We'll see later on.
Okay.
In where these signaling molecules are acting on.
Okay.
Okay? And then with this cascade effect, this domino effect will go all over our body and will produce a resilience to stress. So we'll be more resilient, we'll be more healthy, and we'll have like an overall wellness. That's why when we hear testimonies from different people, they say different things, different benefits when they take Asea, when they take redox, or when they use Renew 28. It's because these molecules, because of the cascading effect, they will produce an effect where the body is wanting it, where it's necessary. So this is just to illustrate how these molecules will work. We take it and because we know the mouth, inside the mouth is a mucosa, and mucosas usually absorb things very fast or faster than the other tissues. So we keep Asea redox, or I'm gonna call it ARS, okay? From now on, it's easy for us to understand Asea redox signaling molecules. So that's why we keep ARS, the liquid in the blue bottle, in the mouth for 15 to 30 seconds, so the molecules can be absorbed, and whatever is not absorbed in the mouth, we swallow it and it will be absorbed later on. Remember that it does not need permission to enter the cell. That's why it's absorbed, and it can be absorbed anywhere in the digestive tract. So from there, the signals are going to be sent all over the body, and it will trigger a cascade effect. So this is just an illustration. Outside the cells, the molecules, this is O2, but O2 is oxygen, it's just to illustrate, okay? So outside the cells, the cell membrane, inside the cells. When we take this supplement, this is what happens. So it's just absorbed. Nothing is needed to help these molecules to enter the cell.
Does stomach acid not kill them? I mean, because you're showing them being absorbed even past the stomach and
No,
everything.
they react with them and they form other redox molecules. Yeah. Okay, the cascade effect, we can visualize it here. Let me see if I don't get it wrong. Don't click too much. Yes, I click it too much. Okay, so it's like when you take one, oops, sorry, let me go back. You take a sip of ARS, leave it in the mouth, one cell is stimulated, okay? This one cell will trigger effect, and the cascade effect will start. and will signal the next cells, the next cells, the next cells, until all of the cells of our body is can be reached by this effect. Okay?
So do you always need to keep it for a few seconds in your mouth? Or
Yes.
just straight?
Yes, you can keep a few seconds in your mouth. Okay, so what do we want with this? We want our body to experience homeostasis. Homeostasis is a balance. We need our body to be balanced and to be more resilient. If our body is balanced and it has the right amount of molecules and also protective proteins, we'll see that we'll be more resilient. Our body will face stress, will experience stress, but it will be easier to go back to normal. This is resilience, right? When you have like a rubber band and you push it to the limit and you let it go again, it will go back to the original form. That's what we want with our body. We want it to be stretched out, stressed, because we all are, right? Almost 24/7, we are under stress. Doesn't really, not only mental stress, but like in contact with pollution, or breathing pollution, or eating something that is not good, or emotions, or anything that could trigger any kind of stress. We want this stress, and we want our body to be able to be resilient and to bring back our body to balance. This is what we want, and this is what we've been expecting with this product. Okay, stress, one side, and then we need a response, and we go back to where we started. So this is, and then we balanced again. This is resilience. Okay. Okay. Beck and Henry will talk more about cell performance later on, but these are the other products that we have, and it's kind of a little bit different, and I'm gonna touch on the points that are different now, and they will talk more about the ingredients and the effects and the benefits of cell performance. Right, Becca?
Mm-hmm.
Yeah.
Okay. So what's the difference between cell performance that I think you were taking it? Are you taking it?
Yeah, we all have them.
energy, yeah.
Yeah.
So these 6 packs right there, what's the main difference between them and the redox sickness? So we have 2 lines of supplements. We have cell performance and we have cell signaling supplements. So cell performance, what are they and how do they work? Okay, they are made with botanicals, vitamins, minerals, and these are big molecules or bigger molecules. Most of them are larger than only signaling molecules. So these large molecules, they will need something else. They are not absorbed by our body. They need help to enter the cells. So basically, they need to be digested, or if you want a more technical term, we can say metabolized by our body. But if you want to understand like general public understanding, we can say that they need to be digested. They need to be broken down. A lot of those botanicals, those ingredients, they will need to be broken down. When after they are broken down, they will go to blood circulation, and then they can reach all of our body. But if you've heard about lock and key receptors and molecules, so molecules have like specific receptors in some cells, in targeted cells. This is how these cell performance products work. They will have a specific target in specific cells in specific tissues. They're not like overall. They're going to blood circulation, but they can stop only where they have receptors from. Okay? So we're targeting specific functions. So we have different active ingredients that we're going to ingest them, and then they have to be broken down into other compounds, blood circulation. It will be transported through our bloodstream, and they will stop where they have specific receptors. So they have specific targets. That's why we say that cell performance is a targeted supplement. So we have redox energy, radiance, mind, and mood that will play a role, will function in specific areas of our body, specific functions like producing energy, skin, mind, focus, or in the case of redox, mood, calmness, and well-being. These are more specific supplements that will work in a targeted way. Is everything okay so far? Mm-hmm. Yep. Okay, this is the comparison of both of them. The supplements that we have regarding signaling molecules and cell performance. So signaling molecules are tiny molecules, are made of tiny molecules, while cell performance are made of larger molecules. Signaling supplements work through absorption because they're tiny molecules, they can be absorbed. Cell performance, because they have larger molecules, they need to be digested. Here they will turn on and off switches that will regulate somehow the production of proteins. These ones, they can also do that, but they are more specific because they act through lock and key effect to specific target places. That's why they have targeted results, and these have this domino effect that it can go anywhere and do the function wherever our body needs. This will consequently lead to resilience to stress and overall wellness, and this a more targeted wellness. I can need more calm, I need to sleep better, then I take mood, I need more energy, then I take energy. I need focus, I take mind. I want my skin to be shinier, smoother, then I take radiance. Okay? All right, so these are the kind of science that, or the understanding of how the products work, our supplements work. Now let's talk about a little bit about science. The ingredients, the compounds that we have in the cell performance, they are well-known ingredients, okay? But they're blended in a very specific manner to produce the best benefits out of each compound. And Beck and Harry are very good in explaining that. They explain to you that later. I'm gonna focus here on the science of the blue bottle and Renew 28. Okay, those are the redox signaling molecules. So in 2017, Asea, I was not with Asea. Asea had a third party lab, the worker that Asea worked with, called Reoxin, and they hired, or they collaborated with this target lab to run a placebo-controlled study, human trial. So we had 60 participants, and they took Asea for a period of time, and then for 2 weeks, then they stopped taking Asea. Okay, they took Asea, collect blood, do the analysis, stop taking it, collect blood, do the analysis, and start it again, and do the analysis of the blood. What they found out was that these subjects that were taking Asea, the blue bottle, they had some of their genes were upregulated. interesting thing is that when they stopped taking Asea, the upregulation went back to normal, to baseline. That's how we call. So we had like upregulation where when they were taking Asea, stopped taking Asea, went back to normal. Okay, and so they also found out that they had increased transcription in some genes, and this is where we want to focus in which genes the Asea supplement acted on. So these are the genes that they found there was significant change in gene expression. So these are just name of genes, right? What do they do? Why are these genes important? So we have 5 genes that were modulated by Asea supplement, by ARS. Remember that the subjects took Asea, had their genes modulated, upregulated, stopped taking Asea, it was back to normal. So how important are these genes? So these genes, they are part of at least 15 different pathways. These pathways are related to different kinds of functions in our body, from serotonin to immune system to interferon beta to thyroid to NRF2, hormones, different kinds of pathways. These pathways have their implications and play certain functions in our body, but we had to decide to go deeper in one of them. And we decided to go deeper in the NRF2 pathway. Why did we decide this pathway and not all of the other 14? Because this pathway is directly related to oxidative stress. And this is how our body builds resilience. By being resilient to oxidative stress, we can balance resilience. And that's how we move to overall wellness. Okay? So NRF2 is, we call master switch, you know, the lamp switch that we turn lights on and off. That's what this molecule do. It turns on and off genes that are related to oxidative stress, that will protect our cells from oxidative stress. We call protective proteins, and that's what it does. That's why we choose also to work with NF2 in the first moment. So this is the definition and why this is important. And we already talked about it. It's a switch, a principal switch. It turns on and off genes. These genes are directly regulated by NRF2. So this is the name of a protein, okay? And there are some genes that are directly related and will only be expressed if activated by NRF2. And the response of the activation of the genes is the production of those protective proteins. Okay? Are you with me still? Mm-hmm. Okay. Okay, so we partnered with 2 different universities. University of Bath in UK and Western Sydney in Australia. Bath, we studied Renew 28, and Western Sydney, we studied ARS, the blue bottle. And here's the interesting results. Okay, remember, let me go back a little bit. I said that when we stimulate our cells, we activate NRF2. NRF2 will activate certain genes where they produce certain protective proteins. One of these proteins is called glutathione. Have you heard about it?
Yes.
It's a buzz right now, right? There's a lot of, even the pure glutathione, you can buy it on Amazon, for example. Okay? So when this study was conducted in monolayers skin cells, so in vitro, using skin cells, and when these cells were cultivated in the presence of ARS, they figured out that there was an increase in 40% of in the production of glutathione. So
is it in the production of the glutathione or the uptake?
production.
Okay, so 40% increase in production.
The skin cells,
Intracellular.
yes, the skin cells were producing glutathione.
Okay.
Okay? More interesting, they blocked the production of glutathione in these cells. And then so cells were not producing glutathione for any reason. Okay? Then, still with the blocker in there, they added Renew 28. What happened is that these cells were able to restore the production of glutathione, even with the blocker in the medium. Because when you cultivate in Petri dish, we have to have a medium so the cells can live in there, okay? So the blocker was added, and then no glutathione was produced, and then Renew 28 was added. Glutathione production was restored by 55% compared to control.
How common is that?
How come?
You know how common?
How common is
How
that?
common?
How many other materials have ever been tested and do anything like that?
Oh, we had tested only the skin cells.
I
so
mean,
far.
across the world, anything else?
Oh, not with Renew 28.
We haven't seen
Yeah.
really any other products that can do what our products are doing.
That's what I was trying to get her to say.
There is well, actually, the scientist didn't disclose that to us because she also can't say if there's any other products that she's been researching with did that. But this is amazing because if we take it to real life situation, what does that mean? Our skin is the larger organ in our body, right? And it's the first contact with, we can even say that it's a big immune organ, right? An organ of the immune system. It doesn't produce immune cells, of course, but it's the face that we have with the environment is our skin. So everything that is bad, it goes through skin. From the air, pollution, and insults, anything, first step, skin. Even things that we eat will have an impact in our skin. So what does that mean? It means that if we're exposed, for example, to a certain pollutant, and for some reason it stops the production of glutathione. Glutathione, remember, is a protective protein. With Renew 28, this production can be potentially restored. So in real life, it means that Renew 28 might be able to restore the production of glutathione, which is a protective protein, when our skin faces an insult, a stress. Okay, and this is very amazing. We're really happy with the results. And more than that, we went even further on. Let's understand this picture here. Okay, this is the cell. These are the cells in culture in Petri dish. like culture. And here we can barely see, but the cells are here without any kind of dye. They're not staining. This is how they really are when they're in culture. This column here, and we can see a lot of green things. The green color means NRF2. So we stained the cells, and it's a very specific dye that will bind only to NRF2. Okay, so everything that is green here is NRF2. Do you remember what NRF2 is? You can tell me. What?
What's the pathway?
It's one of the protein pathways that you
Yes, and what does it do? It turns?
It turns on the switch
Perfect.
to reduce oxidative stress.
Yes, to fight oxidative stress, right? Very good.
It's a molecule?
It's a molecule, yeah, it's a protein.
Oh.
It's a protein, that's the
Yes.
thing. I was like, well, how does a pathway light up? Because it's a protein,
It's a
and
protein.
the protein is a pathway?
A pathway is a set of different proteins. Like we have protein X, and then will act into the protein Y, that will act into the protein A, B, and C, that goes into the nucleus. This
Okay.
is a pathway.
Got it.
And will trigger like gene expression into the DNA and so on.
And so one pathway is this NRF2 pathway. And that's
Yes.
what you're lighting up there, is that particular pathway.
Exactly, okay? And this is the protein. This is the NRF2 in green.
Okay.
Everything that is here is NRF2. NRF2 has something very interesting, and it's even already, someone,
Tyler drew that for us.
did that. So we have NRF2 here, and it's coupled with another protein that's called KEAP1. I think this is bad, huh? Let me try it here.
Yeah, Tyler was talking about keep one this morning.
Okay. So I think if I draw, it will be easier. So we have, if here is, so here's the cell, okay? Here's the nucleus. I'll put the DNA here so we all know that this is the nucleus, the center of the cell, where everything happens when we keep our genes. So, in this place here called cytoplasm, we have NRF2 coupled with, let's do this, coupled with another protein called KEAP1. When NRF2 is like that, it plays no function, it has no function. It is inactive, okay? To be activated, NRF2 needs to be released from these proteins here, okay? Few things can break this binding thing here. One of them are the redox signal molecules, okay? So, when this is broken, when this binding is broken, NRF2 is released and it moves to the nucleus. And when it moves to the nucleus, it plays a role there that will act on certain genes that we call antioxidant response elements. These genes will produce, for example, glutathione. Okay? So, you asked about pathway. This is a pathway. Okay?
What was ARE stand for again? Antioxidant
Response
Response.
elements. Certain genes that are responsive only to NRF2. Okay? Alright. Too much science?
No.
Still here? Okay.
It's a lot of science, but it's very interesting.
It's interesting? At least, okay. So let's go back here. So the blue thing is the nucleus, this part here. Right?
Mm-hmm.
So we dye only the nucleus. That blue stain is nucleus. The green stain, again, is NF2. So there are a lot of NF2 everywhere. Okay? But we want to know if the NF2 is activated. So where is it when NF2 is activated? Where is it located again?
The beginning when it's broken.
Here or here?
No, there.
In the new.
Nucleus.
No, in the
It's the nucleus.
cytoplasm.
Here it's inactive
Oh, okay.
because there's this binding here. So when redoxing molecules come here and break the binding, NF2 can go to the nucleus and activate the protective genes. Okay, so we want to know if our product is able to do this, to break here and send NF2 to the nucleus. Because we know that it stimulates the glutathione production, okay? Which is this. But we want to know the pathway. That's the reason why we did this staining here. Again, NF2 all over the cell in green. Blue is the nucleus. So we have two information. How do we put them together to answer my question? We just overlap the pictures. Okay? Overlap this and this one. Put one on the top of the other. Okay, we have kind of an app, or how do you call that? Not a device.
Like a
A
program?
program, thank you. We have a program that does that. So we take the picture in a dark field, stain, and then overlap both images. Can you see that here? What color is the nucleus here?
Blue.
Blue. And you still have a lot of green outside, right?
Mm-hmm.
What does it mean? It means that the green outside, there is a lot of NF2 bound to keep one. First row. Second row. What is the difference between this, the color of this nucleus, and this one?
It's much lighter.
Kind of aqua blue, right? What does that mean? It means that these two images were overlapped, and this aqua here means that the blue overlapped with the green, meaning that the NRF2 is in the nucleus. It means that our product broke this bound here and translocated NF2 into the nucleus. That's what that picture means. Okay, we treated our cells with redoxing molecules, Renew38. They were able to break this binding here. NF2 is free now, and it can go to the nucleus. And now we know that this glutathione that was produced is due to activation of NF2. Okay? Everything. And this is just the percentage. This is the graph of the picture. I know it's easier to understand the graph, but the picture is more beautiful. Agree? Yeah, right? So this is fluorescence, and cells treated with Renew38 is the quantification. How much NRF2 was translocated, was moved to the nucleus. Control compared to Renew38 treated cells. Okay? How much time do you have?
It's 3 o'clock.
Let's go. Let's get going. Okay.
Or it's 2 o'clock.
Yeah, we're almost done.
Yeah.
So next study we did with ARS. It's in the Western Sydney University in Australia. And what we did, we treated cells, still in vitro, so monolayers of cells. We treated them with Asea redox signaling molecules with our blue bottle. And what we found out was that NRF2 was activated by Asea redox and increased the activation by 60% compared to control.
That's huge, Andrea, because to put that in perspective, the company I've seen that is closest, and they literally built their whole company on NRF2 production, whatever, it was only at 100%.
Yeah.
And we're over here at 160%. So we're in our own class right now. That's for sure.
And our next step now with this university is to do the same thing that we did here. Here we did with glutathione in skin cells. And now we're going to do in different kinds of cells, also glutathione, but for other types of protective genes that are part of these genes that are activated by NRF2. So this study was a really interesting study because we initially sent the blue bottle to them, they tested, and then they're like, not sure if the results are clear or correct. Let's test it again, because it's just salt and water. And they tested again, and they saw the difference. And they were like, not sure again. Let's test it again. And they tested again, 9 times, because each test has to be 3 different sets of Petri dish cells. Okay, so they tested 9 times. And then they're like, okay, we kind of believe now, but they didn't charge a cent more for the other 2 tests because they didn't believe the results, so they want to double check. Not our problem, right? We didn't have to pay, the results are there. And then they were like, okay, we'll send to another lab in the university. And this other lab in the university will test, because we need to be sure that we are talking the right thing. And they sent to another lab in the university, other personnel, other people testing ARS, and they saw the same thing. So they tested like 12 times because they didn't believe the results. But the results are there. So how can we question, right? We're on the top of it. In Australia, the project was good, and so good that we got a grant from the government to run the project. So half of the project was paid by the government. Asea paid half, and the government paid the other half, because the project was really good, and the initial tests were really interesting. So it is.
Was that the Australian government or the U.S.?
Australian government. Yes. Okay.
Something interesting on that, Andrea, so your control is just salt and water, right?
Yes.
So a lot of people that say we're just salt and water, if we were just salt and water, we would get the same results.
And that's why they want to test like 12 times.
Yeah.
Because the content, we have to disclose the content, right? The content in the bottle that we use to produce is salt and water. But we electrolyze. Remember that one of the first slides, and we produce the signaling molecules. So now we have signaling molecules doing this, and the control, as here said, is salt and water. What we do is, remember the graph? When we dilute water and salt, and then it goes to electrolysis, right? Water, salt, we take a sample, and this is the control. Exactly the product before electrolysis is our negative control, or control. Okay, and then the electrolyzed water. So In summary, what are our results from NF2? That our results show that redoxing molecules increased activation of NF2 pathway. NF2 pathway is a very important protein that combats oxidative stress. And how it combats oxidative stress? By producing antioxidant proteins. And this pathway, these results are produced thanks to our Asea redox supplement and Green 28.